where is a given messenger RNA? Is it in the cytoplasm or in the nucleus? How much of it is there? And in the context of aging, we are interested in understanding how our cells respond to stress. By comparing how a young cell responds with how an old cell responds, we're getting very useful information about what it is that we lose as we age. What are the types of responses that our body is no longer able to mount properly as we grow older. So the stress that we have used to generate the image that you see here is arsenite. Arsenite is a very strong oxidant, and oxidative stress is one of the stresses our bodies have to cope with during our lifetimes. So when we treat these cells with arsenite, something very interesting happens, and that is that the RNA in the cytoplasm goes to these distinct foci, cytoplasmic foci, and we know it's a cytoplasm because it's outside of these round, larger circles that represent the nuclei. So now the RNA is assembled in these foci that we call stress granules. And for this image, we used antibodies that recognize two proteins called HUR and TIA1 that we know, based on past work and work of other labs as well as our own, we know that HUR and TIA1 go to stress granules, and there they retain the messenger RNA until the stress in the cell has resolved and the cell has fixed the damage that took place because of the arsenic treatment. And these structures that look very nice and distinct will actually dissolve and disappear. So if we came back 2 hours later, we would see none of these stress granules. They would be all dissolved into the cytoplasm. I think the NIH is very unique in that you have, for example, the NIH is a good example. You have an institute where people with very different skills, very different approaches to science, very different study model systems, they all have in common the same interest in understanding the process of aging. So we have expertise in cell and molecular biology, but we can collaborate with colleagues down the hall who work with animals, and then we can sort of ask the same questions in an animal model. And then we have colleagues who work with patients, so we can ask the same questions that are asked here in a very basic, on a very basic level, we can ask in clinical samples.